This video tutorial is all about the link reaction and the Krebs cycle. Firstly, we're going to focus on pyruvate. Pyruvate, produced during glycolysis, is transported across the outer and inner mitochondrial membranes via a specific pyruvate H+ symport, a transport protein that transports two ions or molecules in the same direction. The pyruvate is then transported into the matrix. In the matrix, pyruvate is converted to a 2-carbon acetyl group during the link reaction. The acetyl group is then oxidized during the Krebs cycle. First of all, we're going to focus on the link reaction. The link reaction occurs in the mitochondrial matrix. Pyruvate is decarboxylated and dehydrogenated, which is catalyzed by a large multi-enzyme complex called pyruvate dehydrogenase, which catalyzes the sequence of reactions that occur during the link reaction. No ATP is produced during this reaction. Firstly, the carboxyl group is removed and is the origin of some of the carbon dioxide produced during respiration. This decarboxylation of pyruvate, together with dehydrogenation, produces an acetyl group. The acetyl group combines with coenzyme A, or the shorthand for that is CoA, to become acetyl-CoA. The coenzyme NAD becomes reduced. Coenzyme A accepts the acetyl group and then, in the form of acetyl-CoA, carries the acetyl group onto the Krebs cycle. A bit like the link reaction, the Krebs cycle takes place in the mitochondrial matrix. The Krebs cycle is a series of enzyme-catalyzed reactions that oxidize the acetate from the link reaction to 2 molecules of carbon dioxide, while conserving energy by reducing the coenzymes NAD and FAD. These reduced coenzymes then carry the hydrogen atoms to the electron transport chain on the cristae, where they will be involved in the production of many more ATP molecules. I'm going to go through the Krebs cycle step by step. First of all, starting from number 1. So the acetyl group, released from coenzyme A, so acetyl-CoA, combines with a 4-carbon compound called oxaloacetate to form a 6-carbon compound called citrate. Number 2. Citrate is decarboxylated and dehydrogenated, producing a 5-carbon compound, 1 molecule of carbon dioxide, as well as 1 molecule of reduced NAD. Number 3. This 5-carbon compound is further decarboxylated and dehydrogenated, producing a 4-carbon compound, 1 molecule of carbon dioxide, and 1 molecule of reduced NAD. Number 4. This reduced or 4-carbon compound combines temporarily with and is then released from coenzyme A. At this stage, substrate-level phosphorylation takes place, producing 1 molecule of ATP. Number 5. The 4-carbon compound is dehydrogenated, producing a different 4-carbon compound, as well as a molecule of reduced FAD. Number 6. Rearrangement of the atoms in the 4-carbon molecule, catalyzed by an isomerase enzyme, followed by further dehydrogenation, regenerates a molecule of oxaloacetate, so the cycle can continue. For every molecule of glucose, there are 2 turns of this Krebs cycle. The table on the slide in front of you shows the products of the link reaction and the Krebs cycle for one molecule of glucose. Although oxygen is not directly used in the link reaction and Krebs cycle, these stages will not occur in the absence of oxygen, so they are aerobic. By the end of the Krebs cycle, the production of carbon dioxide from glucose is completed. Other substrates besides glucose can be used or can be respired aerobically. Fatty acids are broken down to many molecules of acetate that enter the Krebs cycle via acetyl-CoA. Glycerol may be converted to pyruvate and enter the Krebs cycle via the link reaction. And amino acids may be deaminated, so the amino group NH2 is removed, and the rest of the molecule can enter the Krebs cycle directly or be changed to pyruvate or acetyl-CoA.